This is a production of Cornell University.
Thanks, Nate, for the introduction. Thank you, everyone, for coming today. I'm Angela, and I'm going to be sharing some research plans and some preliminary results for my PhD work here, which is focusing on nanoscale characterization of carbon-mineral interfaces, particularly in intact soil microaggregates. So to start on the very broad scale, these are just some values of carbon stocks in various parts of the global carbon budget. And soil, as we know, is a fairly large stock of carbon, especially compared to vegetation. It's commonly stated to be about 3 times the content of carbon in the atmosphere. So this really relatively large stock is important for the overall carbon budget, and whether carbon in soil is increased or decreased or stays consistent is a really important factor for overall carbon balance. So this is just a global soil organic carbon distribution, and from here on I'll be focusing on organic carbon, showing in northern permafrost really high carbon concentrations in blue. But I also like to focus on some more hotspots of high carbon content. In this case, I'd like to talk mostly about volcanic soils in areas like Indonesia, Mexico, and particularly Hawaii. So now zooming in to the island of Hawaii, the carbon content in volcanic soils, which are part of the andesol soil order, have a relatively high carbon content compared to a lot of the other soil orders. And also based on radiocarbon data, a lot of the carbon is very old, meaning that it's fairly stable in soil and has been there for a long time. But this actually depends a bit on the age of the soil. So if you imagine brand new lava deposits just being a few hundred years old, ranging to soils that have been weathering for 4 million years, somewhere in the middle, there's a peak in the amount of carbon content and also carbon age. And this is a concept that has been linked particularly with mineral properties of that medium-aged soil. And this is particularly apparent in deeper horizons. So this is a map of organic carbon content at a depth of about 60 to 100 centimeters, with a red showing higher contents. And relative to, for instance, the contiguous United States, you can see that deep soil horizons in Hawaii and other volcanic soils have much higher carbon content. And again, this is linked with particularly mineral properties in this system. And this is a really good example of the overall relationship between carbon stabilization and minerals. And this is apparent in many different ecosystems with varying degrees of importance dependent on that mineralogy. So this is a process that I like to think about as ranging from the ecosystem scale all the way down to the scale of a few microns, a few nanometers at the actual interface of carbon and minerals in soil. So again, in the example of the volcanic soils, you have an ecosystem property, mineralogy. And in these medium-aged soils, they are dominated by what we call short-range ordered or semi-crystalline minerals. So I'll refer to those kind of as both SRO or semi-crystalline minerals for the rest of the talk. And then moving down to the nanoscale, there are properties at the actual surface of these minerals, such as pH-dependent charge and high surface area, that are related to their carbon storage properties. So I'm just going to give a brief overview of some of the proposed mechanisms of carbon stabilization by minerals. But an important concept is that the actual mechanism involved is very specific to the mineral, the type of carbon present, and also the environmental factors that are at work in that particular system. So just one example is a mechanism of ligand exchange or anion exchange for stabilization of soil organic carbon by the semi-crystalline or short-range ordered minerals that I mentioned earlier in volcanic soils, allophane and ammogalite being 2 examples of that kind of mineral. In addition to ligand exchange and anion exchange, just a few other general interaction mechanisms, hydrophobic interactions, hydrogen bonding, cation bridging, general effects of entropy, and then a little bit less direct than actual surface stabilization. You can also have more indirect effects of physical protection by mineral facilitated aggregate formation or stabilization in pores. So these are all just very general proposed mechanisms of carbon stabilization by minerals. One of the more interesting models of surface interactions is the zonal model proposed by Kleber, where you have amphiphilic molecules, which basically have a polar end and a nonpolar end, creating this kind of bilayer model where you have the polar end interacting with the surface, and then the long sort of nonpolar tails interacting in a hydrophobic zone, and then lots of potential interactions with the aqueous solution on the kinetic zone. So there are many different proposed mechanisms, but why does that really make a difference? Because if we know it's stabilized, why do we need to know how? And I think that it's really important because the actual mechanism involved may respond differently to changing environmental variables. Increased temperature, increased moisture. In particular, an example would be a covalent or an actual chemical bond, an inner sphere complex formation, for instance, may respond differently to warmer temperatures than, say, a weak hydrophobic association. So knowing what kind of mechanism is involved could be really important for prediction of future soil carbon stabilization in modeling, for instance. Currently, all of these mechanisms that I've mentioned have mostly been determined indirectly by adsorption isotherm experiments, for example. And currently, they lack direct observation and characterization in real soils or in situ. But what we do have so far for analysis of carbon mineral interfaces in soil has shown, unsurprisingly, a very heterogeneous distribution of carbon on the scale of an aggregate and all the way down to the scale of an actual mineral particle. So this image shows carbon in yellow, and this is nano secondary ion mass spectrometry. And you can see that the carbon is really distributed in discrete patches and not distributed evenly over the surface of the mineral. And this patchy distribution has also been observed all the way down to a single mineral surface. So this presents a pretty substantial methodological challenge for observation in terms of both the spatial heterogeneity and also chemical variability. So to get a little bit more into the methodological challenges, namely spatial resolution is a huge limitation. So some current techniques, such as NEXAS, which is near-edge X-ray absorption fine structure, which is a synchrotron-based technique, has about a 25 to 50 nanometer spatial resolution, which is just too big, unbelievably, it seems so small, to encompass the kind of variability that we see in soil. Also, some other methodological challenges are the use of a stabilizing material to actually make thin sections, which is often carbon-based, so then it's really hard to see carbon when you use a carbon stabilizing resin. Also, for instance, in this study, mineral particles were kind of shaken up and then distributed on a surface, so you don't maintain the spatial distribution in an actual aggregate. And then also, in general, high energy analysis using an electron beam or X-rays can create sample damage. So these are all things that have to be considered for direct observation. So one method, this is now what I will be focusing on for the rest of the talk, is a combination of scanning transmission electron microscopy, or STEM, and electron energy loss spectroscopy, lots of acronyms, EELS. So just really briefly, EELS involves the electron beam from the microscope going into a very thin cross-section sample. And then some of those electrons will just pass right through, and some of them will interact with the material and then scatter. And so some of the scattering is inelastic, which means that it loses energy. And so you can measure that energy loss with a spectrometer, hence getting electron energy loss spectroscopy. And that's just basically how that works. And then when you get an energy loss spectrum, you have some distinctive peaks for certain elements. So this is for carbon, which is in diamond and graphite. But then you have a distinctive peak position and shape, or the fine structure, which tells you something about what that carbon is bonded to. So if it's bonded to oxygen, it's a double bond to another carbon. Nitrogen, you can get an idea of what functional groups are there and what their coordination might be. This is an image of stem eels of a Amazonian dark earth that has biochar particles. But this particular image shows an aluminosilicate clay in red and then iron oxide on the surface. And you can determine using eels that it's oxidized iron, or iron 3, based on the intensity signals, which I'll be talking about later. So eels is a really good tool for both very high resolution, ideally for a homogeneous material. The resolution can be up to 0.2 nanometers, or about 2 angstroms, which is unbelievably high. For natural soils, probably not that good, but that's kind of the ideal. And then also be able to get information about the chemical composition of the sample. So it's a really useful combination. So now I'd like to give an overview of the main chapters or sections of my PhD research. So to start, I'm going to be compiling a library of sorts of eel spectra for a number of different soil minerals, and then also common model carbon in soil. And then building on that, collect eels data for simplified mixtures of carbon and minerals. And that will be designed based on specific hypothesized interactions that have interesting spatial information. In this case, a simplified system where the environmental variables can be controlled, so redox potential or pH, that somehow may be affecting the interaction mechanism. And then sort of the end goal, or the epitome of all of this, is applying the technique to real soils in thin sections, while, importantly, maintaining spatial distribution in the sample, eliminating the use of stabilizing materials, or carbon resins that would allow us to not be able to analyze carbon, and then also minimizing sample damage. So I'm kind of pursuing each of these three things all at once. So I'll show you a few aspects of each of them, and then the next steps for each as well. So to start, some of the standards that I've chosen are listed here, and they represent a range of elements. I specifically am also analyzing volcanic soil minerals, allophane and ammogalite, for instance, as well as some other elements, calcium and phosphorus minerals. And then similarly for carbon, there are many different kinds of carbon present in soil, as we know. And so I've selected a list that cover a broad range of functional group types, and also some that are specific to black carbon, or biochar, because I'm in Johannes Lehmann's lab. So I'd just like to show you a couple of reference spectra that I've collected so far. So these are for hematite and goethite, both iron minerals. Hematite is an iron oxide, and goethite an iron oxyhydroxide. So each of these particles is ground very fine, and then deposited, this lacy background is carbon that you just deposit the materials on. And then you end up with some areas on the edges that are thin enough for eels. So the whole particle is going to be way too thick, but you can get some kind of regions on the edges. Before I start showing a bunch of spectra that are not going to mean a whole lot, I just wanted to give a quick overview of some of the terminology. So when you're talking about nexas or eels, you're talking about K, L, M, and N edges. And these basically correspond to the simplified Bohr model of an atom, the energy levels that you can also see in your Gen Chem electron configuration sort of representation. So you have your core electrons closer to the nucleus that are your K and L edges, and those will require more energy to be excited or to move to the next energy level. And then out in the outer edges, you have the M and N, which are more of your valence electrons. So those require much lower energy. This is just a simplified schematic of an EEL spectrum. So right at 0, you have a huge peak called the zero loss peak, which tells you how many electrons just pass straight through your sample. And you want to know that because you want to know that it's thin enough to be able to use this technique. So that's a really important thing to collect for all of your spectra. And then you have this kind of indistinct bump in the low loss region. And so that's indicating all of those M and N edges in your valence electrons. And there's a lot of overlap between different elements in that region. So it's kind of hard to get really qualitative information from that, but you can kind of confirm that, okay, well, I see the iron M edge, so that means iron is definitely present here. But when you start to move out into the higher energy regions, where you might be looking at your carbon, iron, oxygen edges, the elements of interest, that's where you start to get an apparent peak for the core loss, and then all of this fine structure that tells you something about the binding environment of that element. So here is a spectrum for hematite and goethite for iron. So this is the iron L edge now. And on initial kind of observation, they look fairly similar. However, even very small shifts can be meaningful in terms of discerning between different kinds of iron. So if there's a little bit of a difference in the shape of this little shoulder, there could be a shoulder here that could vary by different kinds of minerals. Like this very subtle shift in peak position may be meaningful. And currently, there isn't a whole lot of information in the literature about these fine distinctions between iron minerals and soil. So this could add some really good information to that. This spectrum, so hematite has been imaged before with eels. So this is a spectrum in the literature, so it's quite similar. And also, which has been pretty well established, is that the ratio of L3 and L2, these two peaks, is a function of the sample oxidized to reduced iron content. So that's really convenient, and that's how we can use eels to kind of discern iron 3 and iron 2. So there's a pretty consistent relationship between the ratio of those peaks. So you could have hematite, which is theoretically 100% oxidized iron, and then something like phthalate, which is theoretically 0% oxidized iron, and be able to kind of map in this spectrum a wide variety of different redox states in iron, which is really a useful tool. For oxygen, you can see that this spectrum starts to look a lot messier, which really takes away a lot of the conclusions you can draw from it. But there is a pretty apparent difference between the ratio of A and B in this spectrum. And it turns out that that is related to the degree of oxidation. And this is also a very consistent, predictable trend. So you can use that to determine the iron-oxygen ratio, which is another really useful tool. And finally, for hematite and goethite, this is the valence region, the really low loss. So starting just above 0 up to about 60. And so there isn't a whole lot of really great information here. You have carbon coming from the grid that we prepared the samples on, but you do have an iron M edge here. So that's really good for as like an independent confirmation that iron is present, and we might be able to get some information from that as well. So just quickly, two other minerals that we've analyzed so far are gibbsite and hydroxyapatite. So looking at aluminum, calcium, and phosphorus. Aluminum is really hard to get EEL spectrum for because it is in the low loss range, and as I mentioned, there's a lot of overlap there. So that's a challenge, which is a little unfortunate because aluminum, of course, is very common in soil and a very important part of mineral interactions. And just quickly, this is a list of the remaining elements, or the remaining minerals that I have to analyze. As I mentioned, some volcanic minerals, allophane and amogolite, some kind of normal crystalline clays, and then also iron oxides that represent that range of iron oxidation. And we have started working on some carbon standards. An issue with that is they're much less resistant to damage from the electron beam than iron minerals. And also, typically, when you have any sample that goes into the microscope, you have to clean it with a plasma, an oxygen plasma, before it goes in so that it doesn't contaminate with just things floating around in the microscope. But of course, these are carbon compounds, and cleaning them with plasma would kind of just burn them away, which would be kind of useless. So we don't have a good way to clean them, and so this ends up posing a huge problem with carbon contamination. So in order to get around that, we're working on doing eels at very low temperature called cryo-eels, which helps eliminate carbon contamination, and then also using alternative sample cleaning. One example is critical point drying. So next, I'm just going to give a quick overview of my proposed work in these simplified mixtures. This isn't something that I've started yet, so I'd be glad to hear any input you might have in this or suggestions. And so basically, these are going to be simplified systems in a kind of a classic adsorption isotherm type approach based on some specific hypothesized interactions, and then followed by analysis of the particles themselves with eels to see if we can kind of really unequivocally determine what is going on at that interface. So this is just one example of one of my proposed combinations. So this would be using a polar carbon, shown here just generically as RCOOH, and a nonpolar carbon with a generic carbon chain, and amogolite, another, again, one of the volcanic minerals. So one hypothesis is that the polar carbon will be involved in covalent bonding, but only at the edges or the ends of the amogolite tube or at broken sites on the surface, and that the nonpolar carbon may not interact at all, or it could be involved in some kind of secondary association with the polar carbon. So just to give you an idea of the spatial distribution that we might see, you could see just polar carbon at the ends and nonpolar carbon not associated at all. We could see it everywhere, or just in broken sites, and nonpolar not associated, or nonpolar could go to the ends, or it could cover the whole thing. So there are lots of different possibilities here, but what is great about this technique is that we'll be able to see, a difference between this carbon and this carbon, and be able to assign where it is spatially in this system, which is really useful. And then next, one of the proposed incubations or adsorption experiments would involve a sequence of reducing conditions and oxidizing conditions, reducing, oxidizing, to try to encourage iron co-precipitation with carbon. So there has been some recent work about spatial differences between carbon that's just adsorbed to the surface of oxidized iron versus carbon that's just physically intercalated in the structure of a co-precipitate between iron and carbon. So this is just something that has a really interesting spatial characteristic, which is really the benefit of the technique that I'm using. So it's important to select things that have some kind of interesting spatial information. So finally, I'm going to show you some preliminary results from actual analysis of real soils, which is kind of the ultimate goal of this work. As I mentioned earlier, the main challenges here are preventing damage to the sample, eliminating that carbon resin, maintaining the spatial distribution within an aggregate, and then also being able to analyze both carbon and the mineral chemistry. And so similarly to the cryo-eels I mentioned for analyzing the carbon standards, we're going to be using cryo-FIB, or focused ion beam milling, to prepare the samples, and hopefully this will account for a few of the methodological considerations. And then followed by that, we'll be using cryo-eels, so everything is frozen from beginning to end in water. which is a more natural matrix than carbon resin. And as you might have guessed, my initial soil samples will be from volcanic soils, from a model ecosystem with mineral control on carbon turnover. So just a quick overview of the soils that I'm using right now are collected from Hawaii by collaborators, Katie Grant, who's here, and Lou Derry in Earth and Atmospheric Sciences. This particular sample is characterized as a Waimea silt loam, and it's about in the middle range of soil age, around 350,000 years old. And so it's characterized by those short-range ordered or semi-crystalline minerals that I mentioned earlier. So these are some scanning electron microscope images just of intact aggregates, just to give you an idea what they look like and how variable they are. So you can see that you have some that are kind of lumpy and some that are kind of smooth, some that are stretched out. So even in the aggregate shape, there's a lot of variability. These were collected from deeper soils and also from a system that has relatively high iron and carbon content, and they're well drained, so they're probably dominated by oxidized iron. I selected aggregates in the range of about 53 to 150 micron diameter, so microaggregates. And this is for ease of analysis. It's just a better size to use, and also these microaggregates may have really stable mineral interactions. So this is a little cartoon of how we start to section the aggregates. So first they're hydrated slowly with a humidifier, and then just dropped in a little bead of water, flash frozen in liquid nitrogen, and then cut into sections using a cryo-ultramicrotome or a diamond blade, just a little boop, and then it ends up on a copper grid in a thin section that still has ice around it, and it stays frozen. So then, if you can visualize it, this is now that grid looking from above. So each of these little blobs are sections of carbon, so you can see they're kind of variable. But if you were to look at them from the side, they are actually relatively flat on both sides, amazingly. It's a really interesting technique. And they kind of cover a range of shapes. Some of them kind of break apart on natural breaking points. Some of them look like pork chops, I think. Some of them look like tacos. But then you have to find the one that just looks just right, and that's the one you go with. So then after you have your perfect sections that you want to try to look at, you then, in the fib, which is the focused ion beam, you have a beam of gallium ions that come down at an angle and kind of cut out a wedge out of the sample. So you end up with a really thin area at the bottom, and then just sort of the normal sample thickness at the top. And you can see that in each of these images here, we have our wedges with only the very edge of the sample probably thin enough for eels. So it's getting down to a really small area for analysis, and so you kind of have to get a little bit lucky that you catch a really interesting feature in that tiny area. But we fortunately did last week, and hopefully more to come. So this is just some very preliminary results for some of our initial imaging. So this is just one of those sections, and then a tiny area of one of these sections, then blown up much bigger. And then this tiny area blown up bigger here. And then finally a elemental map of that region. So this is an electron dispersive X-ray, so it's a very coarse scale map. But you can see that we have carbon, which is exciting, and you can see this area here is relatively homogeneous carbon, which is nice. It's not mixed up with a lot of other elements. And then we have a particle of mostly aluminum. It also has oxygen. No silicon, so it's probably an aluminum oxide. However, this is a little bit of a problem. We also have gallium. Where could that have come from? Of course, the gallium ion beam, right? So gallium likes to deposit itself on the sample sometimes. So this is just something we need to be aware of and make sure that it's not changing our signals too substantially. You'll notice, though, that it's really concentrated in the aluminum area and hardly any signal at all in the carbon, which is nice because then we can be able to analyze the carbon without worrying about gallium. But this is something we need to consider for future analyses. So now going even smaller, we have this area of carbon and aluminum blown up to a STEM image, or STEM image. So this is your carbon here. And then each of the areas of different contrast are higher atomic weight elements, so they show up differently. And then even more magnification, we then have carbon, and we can start to see this interface with a mineral that we have putatively determined as aluminum. And then even one more image. This is the highest resolution I have. We have carbon, and then this kind of fuzzy but still kind of distinct interface. So just something to note here, this scale bar is 50 nanometers. So when I mentioned NextS that the resolution is 25 to 50 nanometers, just imagine that half of this or the whole thing is just one whole pixel. So this is a much better spatial resolution already, even at just this scale, than we could get with NextS, which is a really exciting thing. Also exciting, we collected a carbon Kedge yield spectrum from this area. To my knowledge, the first in real soil without a carbon resin, so that was kind of an exciting day. It's not a particularly distinctive spectrum, but there are a few things that even just with very preliminary analysis we can start to see. So there's no peak at 285, which is characteristic of aromatic carbon and very consistent across all types of carbon for NextS, which is mostly related to EELS data. We do have these two kind of indistinct pre-peaks at about 287 and 289 EV. And they could really be a whole bunch of different things. Could be shift of aromatic carbon with an oxygen or a nitrogen, or could be aliphatic carbon. 289 could be carboxylic acids or amines or alcohols. So you can see the value of determining the carbon reference library and analyzing real soils, because we can really start to link these peaks and their characteristics with known functional groups. We also have a line scan, which basically takes points along the line that you draw on the image. So we can start to kind of get a range across a gradient. So first, in the adjacent carbon region, this is the spectrum I just showed. And then starting to get closer to the edge, you can see that the spectrum starts to get a lot messier. Lots of variability here. And that's because it starts to get thicker as you get closer to the mineral. So as we continue to prepare more samples, we really want to try to maximize the thinness of the sample. It's just going to continue to be something to work on. But you can already see that there are some differences potentially in the relationship of those first two pre-peaks just out at this outer edge. And then at a point closer to the interface, there are quite a few new small peaks, lots of variability. This is going to take a lot of analysis to get what might be meaningful out of the spectrum. And then, of course, the less signal to noise ratio we can get, the better it will be at being able to pull out features of these spectra. And then finally, one more on the other side. And you can see that the first pre-peak is almost gone. So the meaning of this is going to take a lot of analysis, but it's kind of an exciting development to see that there are differences across this mineral carbon region. So just to wrap up, the next things that I'm going to be working on: continued sample preparation for the thin sections, continued analysis, and then working on linking carbon compound chemistry and the interface, and then just in general, continuing to analyze standards, the simplified mixture experiments, and then lots and lots and lots of data analysis forever and ever to get meaningful information from this data. So I'd just like to acknowledge my funding, which is from the Cross-Scale Biogeochemistry and Climate IGRT. And then also we've been working with the Cornell Center for Materials Research, which is a really, really essential piece of this. We would definitely not be able to do it without our collaborators in physics, David Muller, who works with EELS, And Lena Fitting-Kirkudis, who does FIB, and then their grad students have collected a lot of this data with me and have been really, really helpful. Also, my thesis committee, my advisor is Johannes Lehmann, who's not here today, unfortunately. And then also we've gotten lots of support from my lab and the support staff at the CCMR. So there are tons and tons of really interesting resources, which obviously you can't read, but if you ever want to talk about where I got any of this information from, I'd be glad to discuss it further. And finally, thank you so much for your attention. Do you have any questions?
This has been a production of Cornell University, on the web at cornell.edu.